In this series of videos on photorespiration, we have been trying to understand how photorespiration or C2 photosynthesis happens. This is a discovery that was made in 1959 by two scientists, Dicker and Teo. These scientists observed that when the temperature is high, if the C3 plants will evolve more of carbon dioxide and they seem to be consuming oxygen. This led to the discovery of this pathway. In the previous video, we looked at the situation that leads towards O2 fixation by Rubisco. We are going to understand how these reactions are going to happen. We begin with ribulose 1,5-bisphosphate. This is the molecule, starting met molecule. Now this ribulose 1,5-bisphosphate, by the activity of the enzyme Rubisco, is fixing an oxygen. And these are 5 carbon. And if oxygen is added, we are getting a 3 carbon, that is 3-phosphoglycerate and 2-phosphoglycolate. Now the 3-phosphoglycerate, the structure is this is 3-phosphoglycerate, and the 2-phosphoglycolate is this is 2-phosphoglycolate. Now we are trying to understand how do we convert this molecule back into the 3-phosphoglycerate so that this can be converted into starch. Now for this conversion, it's a laborious process that involves 3 organelles in a plant. The 3 organelles are, it will make use of chloroplast, it will make use of peroxisomes and mitochondria. These are the 3 organelles that will be made use in order to convert 2-phosphoglycolate into 3-phosphoglycerate. Now what we will do is we will begin with this molecule and we will start with what all happens in the chloroplast. Since the body is small, we will continue to the reaction that will happen in peroxisomes, then we'll continue with the mitochondria. We begin with what happens in the chloroplast. Now in the chloroplast, we have already created 2-phosphoglycolic acid by the activity of Rubisco. Now this 2-phosphoglycolic acid, now if the phosphate group is removed through a hydrolysis reaction that is catalyzed by phosphoglycolate phosphatase enzyme, and once the phosphate group is removed, we will get glycolic acid. All this will happen in the chloroplast. Once glycolic acid is formed in the chloroplast, now in the chloroplast membrane, it has a very specific transporter, and this transporter will allow glycolic acid to diffuse out of the chloroplast. Once it is out of the chloroplast, in the next stage for the glycolic acid is it will get into the next organelle, that is peroxisomes. Now, in the peroxisomes, the entry of glycolic acid is assumed to be through diffusion process. Glycolic acid will enter inside. Now, here is glycolic acid, same structure. Once it is inside, the glycolic acid will be acted upon by another enzyme. The name of the enzyme is glycolic acid oxidase enzyme. Glycolic acid oxidase. So, glycolic acid oxidase will convert glycolic acid into glyoxylate. Structure of glyoxylate is, this is the structure of glyoxylate. Now, in the process of making glyoxylate, it will also produce H2O2. So here, O2 is taken. H2O2 is produced in the peroxisomes. we know that peroxisomes are rich in catalase enzyme. Catalase enzyme will convert this H2O2. So here is catalase enzyme. It will convert H2O2 into H2O and half O2. So therefore, this is how the complete conversion occurs. In order to understand the pathway, what we need is, we have this glyoxylate that is formed in the peroxisomes. Okay. In the next step is very interesting. What will happen is, this glyoxylate will be converted to an amino group is added to glyoxylate. And so the amino group is contributed by glutamic acid. And once we know that once the amino group is transferred from glutamic acid, it becomes alpha ketoglutamic acid. The amino group is transferred there, so therefore it becomes CH2NH2 and COOH. An amino acid is formed, and this is glycine. Glycine is produced in the peroxisomes, and this production is transferred by a transaminase enzyme. Transaminase. So therefore, glycine is produced in the peroxisomes. Understand, we began with the glycolic acid, and glycolic acid transferred to peroxisome, peroxisome converting glycolic acid into glycine. This is the first stage that happens in the peroxisome. Once this glycine is formed in the peroxisome, glycine is transferred to mitochondria. We require 2 molecules of glycine for the next reaction. We begin our story from here. Now this glycine is transferred to the third organelle, that is mitochondria. So here we have glycine. We require 2 molecules of glycine. So therefore, let me write the structure CH2NH2. This is the glycine molecule. If the first glycine molecule will undergo decarboxylation and deamination. So therefore, this is catalyzed by an enzyme known as glycine decarboxylase that will remove the CO2 molecule and the NH2 molecule. CO2 is removed, NH2 is removed. So now if the amino group and the amino group which is removed, generally it is assumed that rather than creating a toxicity, it will be added to a keto acid and it will be converted to an amino acid and it will circulate in the mitochondria. We require a second molecule of glycine. What happens to this CH2 group? This CH2 group is accepted by a tetrahydrofolate. So here we have a tetrahydrofolate. Once you attach this methyl group to tetrahydrofolate, tetrahydrofolate becomes N510 methylene tetrahydrofolate. So the methyl group is attached to this product. We require a second molecule of glycine. So the second molecule of glycine is added here, and now this second molecule the methyl group is added to the second glycine molecule, and then it is getting converted to L-serine. The structure of glycine is CH2OH, CH, NH2, COOH. Now this reaction also requires one NAD plus, we'll write here NAD plus, and NAD plus getting reduced to NADH plus H plus, and this NADH will enter into electron transport chain and ATP molecules will be produced, because all these are happening in the mitochondria. Now once serine is produced in the mitochondria, it will move out from mitochondria into peroxisome, coming back. Now we have serine in the peroxisomes. The serine which is formed in the mitochondria is now transferred to peroxisomes. Now in the peroxisomes, another transamination reaction is going to happen. Serine will be transaminated. The amino group is transferred to alpha ketoglutaric acid, and that gets converted to glutamic acid, and a keto acid is produced. The keto acid is hydroxypyruvic acid. This reaction will take place in the peroxisomes. Once this hydroxypyruvic acid is formed, this will be reduced by hydroxypyruvic acid reductase enzyme, and this is an NADH dependent enzyme. So therefore the two hydrogens are added to this hydroxypyruvic acid. So therefore we have this product CHOH and CH2OH. This is glyceric acid. So all these reactions are happening in the peroxisomes. Now in the glyceric acid from the peroxisome will be transferred back into the chloroplast. Now we have, once the glyceric acid comes into the chloroplast, the last step happens. The last step in the chloroplast. So therefore this is happening in the chloroplast. So therefore glyceric acid kinase, there is a kinase enzyme, glycerate kinase, that will break an ATP, and inorganic phosphate is added to this one. So phosphate will be added here. You will have 3-phosphoglycerate. And this is the 3-phosphoglycerate that is again used for the formation of ribulose 1,5-bisphosphate in the regeneration step, or for the formation of a starch. This is the summary of all the reactions that we have seen. We began from here. This is our chloroplast, where ribulose 1,5-bisphosphate has been converted to glycolate. Glycolate is transferred into, this is peroxisomes. Glycolate is transferred here, which is converted to glycine. Glycine is transferred into, this is mitochondria. And in mitochondria, glycine is converted into serine. Then it is transferred back into peroxisome. Serine is converted to glycerate in the peroxisome. Glycerate is transferred back into the chloroplast, where it is getting converted to 3-phosphoglycerate, and the 3-phosphoglycerate, it can be converted back into ribulose 1,5-bisphosphate, or it can be converted to starch in the process. This is the summary of C2 photosynthesis. This is the overall stoichiometry of C2 photosynthesis. Here there are totally 10 carbon atoms, and it is ribulose bisphosphate. So therefore there are 4 phosphate groups, that is giving rise to 3 into 3, 9 carbon atoms, and 3 phosphate groups, and this is 1 carbon atom, and we need 1 more phosphate, and this is 1 phosphate. So therefore the overall equation is balanced in the stoichiometry. Now we look at certain important aspects of this C2 photosynthesis. Initially we began by saying that we are not seeing any positive aspects in this C2 photosynthesis. In the latest research that has been carried out, several proof has been given for some of the advantages of C2 photosynthesis. Advantages like the NADH which is being produced is utilized for ATP production. This is an effective way for removing the toxic substances, and this pathway is also being utilized for assimilation of nitrate and its conversion into nitrate. And these are some of the advantages that has been proposed for this particular pathway.